Hi, everyone, and welcome back. So in this video, I'm going to be talking about how cells repair mutations. So in previous videos of mine, I've talked about the different types of mutations and how those mutations can impact at the DNA level, the impact that they could have at the protein level, and also discussions about how mutations can impact the ability or the reproductive fitness of an individual. Here, what we're going to talk about is how cells are able to repair this damage in some situations. So let's get started. The first thing we'll talk about is where do mutations come from? So mutations can come from several different sources. There are endogenous sources. So these are sources of DNA damage that occur from somewhere inside of the individual or inside of the cell, endogenous, okay? The first and most common are actually replication errors. So the process of DNA replication, so DNA polymerase and all that stuff we talked about during DNA replication, errors do happen during this process. DNA polymerases do have a proofreading capability, and often they're able to realize when they've made a mistake, and they'll actually pause, go back, and repair those. But in other cases, they just don't catch it. And that can result in the incorporation of an incorrect nucleotide or maybe an insertion or deletion. We'll talk about how that happens later on in the video. Another endogenous source of mutations are cellular metabolism. So we've talked about if you've had classes about in general biology where we've talked about metabolism, one of the things you know is that oxygen in many organisms, in aerobic organisms, is the terminal electron acceptor, which means after the electron transport chain, electrons are given to oxygen to make water. But sometimes that process doesn't happen perfectly, and sometimes you get these things called reactive oxygen species. These are things like superoxide, hydrogen peroxide. They are these, essentially what you have are oxygen species that are carrying extra electrons, and these can actually damage DNA, as well as other parts of our metabolic processes can actually damage our own DNA. So there's a reason why we have enzymes in our cells to help detoxify these harmful oxygen products. But again, it's not always perfect, and this can lead to DNA damage that needs to be repaired. There are also exogenous sources. So these are sources that come from the outside. So ionizing radiation is one of them. So if you're exposed, like, for example, to high doses of nuclear radiation, highly problematic. UV light exposure. So the sun, right? Those UVA, UVB rays, the reason why we wear sunscreen, the reason why tanning is not a good idea. These can actually cause different types of DNA damage, as well as chemical exposure. So exposure to things, basically carcinogenic chemicals, things that are found in like cigarette smoke, alcohol, things like that can also damage our DNA. And as you know, the DNA holds all the information that a cell needs in order to be able to function. So if you're damaging the DNA, this can have a huge effect on the way the cell behaves, right? And I think the biggest thing we're all afraid of when we talk about these is we think about cancer, right? When a cell begins to divide sort of without the reason for dividing, a cell that divides uncontrollably can lead to tumor formation and can become cancerous if not checked. Okay, so in terms of replication errors, there are many different kinds. So replication errors can occur when DNA polymerase incorporates the wrong nucleotide. Occasionally, you end up getting additional nucleotides during replication. So we're talking about things like AG-TC mismatches, right? So we know that T's and A's are supposed to be base paired, G's and C's are supposed to be base paired, but in certain situations, we can end up with an inappropriate mismatch. Again, DNA polymerases do have proofreading capability, and often they catch these things, but at times they can slip through, they can make it through, and these will need to be caught during proofreading or through some of these error repair mechanisms that we'll talk about in a little bit. Another thing that can happen that leads to insertions or deletions. So I've talked about these in my mutations video. We call these indels. At the protein level, they tend to lead to frameshift mutations. But one of the things people don't always understand is how do these insertions and deletions actually happen, right? Why would it make a mistake and be like, oh, I'm just going to leave that nucleotide out? The reason why this often happens is it happens in regions that are highly repetitive. So you can see in this scenario, there's a strand of 5 T's, okay? Which means you should have on the complementary strand 5 A's on the other side. But because you have so many T's in a row, sometimes that newly synthesized strand can like loop out and four of the T's will be aligned. The fifth one won't be. And that results in that A just sort of not base pairing, and it kind of gets like basically they go, oh, okay, I've got right here, we've put three A's in, right? Or four A's in, but only three are connected. So it's going to add an extra A to fill out that sequence of five. All of a sudden now this daughter strand has an extra A in it. That A is now inserted inside. Okay. So if it's the newly synthesized strand that loops out, you're going to get an insertion at that point, because that new daughter strand and all daughter strands that result from that as a template are going to carry that sixth A that looped out and got incorporated. And this happens in repetitive regions because there's still enough base pairing that's occurring where the cell doesn't quite recognize, like, oh, I put an extra A in, because as far as it's concerned, it's got five A's and T's lined up. It's just kind of ignoring that sixth A that's kind of hanging out there. In the other scenario, this is what's going to lead to a deletion, is where you get a looping out of the template strand. So you can see the template strand loops out, and as far as the DNA polymerase is concerned, there's only four T's in this stretch, so it's going to synthesize four A's to complement that, and as a result, the daughter strand is missing one of those nucleotides, okay? And this is, again, because the presence of similar nucleotides kind of makes that okay. As far as that DNA polymerase is concerned, I'm only seeing four T's, so I only need to put in four A's, but in reality, there's a fifth T that's not being complemented. And now you end up with an insertion in scenario one or a deletion in scenario two, and that can have a profound impact on the resultant protein if this happens inside of a protein encoding gene. And by the way, once this happens, it now becomes permanent. That's not going to get passed on, because every single cell that has one of these daughter strands that's either incorporating an extra nucleotide or missing that fifth nucleotide, all of the resulting daughter strands from subsequent rounds of DNA replication are going to carry that mutation forward. So what are the repair pathways that are available to catch these replication errors? Okay, so the first one is DNA polymerase proofreading. All right, so like I said, DNA polymerase is going along and it's incorporating appropriate base pairs. It sees a T, it pairs it with an A. It sees a C, it pairs it with a G. It sees it with an A, and it pairs it with a T. And right here, let's say it sees a G, but it pairs it with a T. Well, now that's a problem, right? So what's going to happen is the DNA is going to kind of bulge out a little bit because you're not getting that base pairing, right? It's almost like if you have a piece of tape that gets a wrinkle in it, you're like, oh, that's not smooth. I've got to smooth that out. And DNA polymerase goes, that's not right. That doesn't feel right. So the back half of the enzyme recognizes there's a problem. It causes the polymerase to stall. It essentially uses its exonuclease activity. So exo means remove. It has 3' to 5' exonuclease activity, which means it can work backwards, right? DNA polymerase only writes 5' to 3', but it can back itself up, remove that mismatch base, and then put the appropriate nucleotide in, okay? So that's what happens. Now, the other thing that can happen is, let's say that DNA polymerase's proofreading capabilities don't detect this mismatch. So here you can see in the template DNA and the new DNA, you've got a GT mismatch, okay? So there's a mismatch on the newly synthesized strand. So what actually happens with mismatch repair is a set of enzymes is going to come in. It's going to recognize that there's a mismatch because the shape of the DNA is wrong. It's going to affect the way the DNA twists. It's going to affect, there's going to be a bulge in that DNA. And it's going to say something's wrong here. And what actually happens is the newly synthesized DNA strand, so not the template, but the daughter strand, it actually just cuts a whole segment out. It doesn't say like, oh, this is a problem. It quite literally cuts this whole chunk of DNA out and then replaces it with a new one. So another DNA polymerase comes in, redoes the synthesis process, incorporates the correct nucleotide here. And then you've got that little nick, those two little nicks on either end where the backbone's been cut, and then DNA ligase is going to come in and seal the DNA backbone. So this form of mismatch repair is very, very similar to what happens when primers get removed. So we talked about during DNA replication where you have RNA nucleotides in there, and those have to go, right? We can't have RNA mixed in with our DNA. It's very much the same thing where they're going to cut out that whole chunk of RNA and then put in a newly synthesized version of DNA to fix that and then seal that gap. This is essentially road construction, right? You've got a stretch of road that's kind of beat up. You start a little bit before it's bad, you end a little bit after it's bad, and you put a whole new set of road in there, and then you've got to make sure it's level at either end. And that's what's happening with mismatch repair. So how does mismatch repair actually work? All right, so first things first, we've got to have replication occurring. Replication happens, and now we're going to end up with some type of mismatch in the region. And they're showing that here with this GT base pair. What happens is, first and foremost, the DNA needs to, or the cell needs to identify which strand is actually the daughter strand, right? So you have to think about this. You've got two strands of DNA. One of them has been newly synthesized, one of them hasn't. It's not like they have name tags on them. So the cell first needs to figure out what's the wrong nucleotide, because you've got a GT mismatch. Which one's wrong? Is a G mispaired with a T, or is that T mismatched with the G? Which one is the template? And that's important. Now, these mismatch repair enzymes first need to identify that daughter strand. In some species, it's fairly easy. In gram-negative bacteria and some other species of bacteria, the parent strand is actually methylated. Okay, so what that means is, after the cell synthesizes its DNA, after all the repair processes are gone, and the cell's like, okay, I'm now a new cell and I'm going to function, all of that DNA gets methyl groups added to it. But newly synthesized DNA doesn't have it. So all it has to do is go, are you the methylated strand or the unmethylated strand? Whichever is in the methylated strand, they know is the template, and anything else that's non-methylated is the daughter strand, so that's the one that needs to be repaired. But in other species of bacteria and all eukaryotes, we're not actually fully sure how cells know which is the daughter strand and which is the template strand. We really don't know that. And we think that it may have to do with some small nicks being present in the daughter strand that aren't found in the parent strand, but we're not entirely sure. The first thing that's going to happen then is an enzyme called Mut-S, okay? Mut-S is going to recognize, yup, there's a problem. And it's probably because the DNA is shaped wrong, something's going on. At this time, it will also try to identify which one is the parent strand and which one is the daughter strand, okay? Then a second enzyme called Mut-L is going to come in, and that's actually going to cut the DNA. So remember, we saw that it's going to take that daughter strand and it's going to remove a whole chunk of that DNA and then resynthesize it. RPA or RecJ, these two proteins come in, and they're going to, it depends on which version you're in, okay? It's going to come in and it's going to remove that whole section, all right? So Mut-L cuts it, RPA or RecJ actually physically removes that, and now you've got a strand of mismatched or unpaired DNA, right? So you've got that non-duplex DNA at that point. At this point, depending on which enzyme, which type of species you're in, if you're in a eukaryote, RPA is going to remove the mismatch, and DNA pol delta is going to come in and synthesize that new stretch. If you're in bacteria, it's RecJ removing the mismatch, and it is DNA polymerase 3 that's going to synthesize this. And you can see right here, Mut-S alpha, Mut-L alpha, Mut-S, Mut-L. Over here on the right, you have majority of bacteria. Over here on the left, you have the majority of eukaryotes. So you can see the process as a whole is highly conserved. The process is going to happen the same no matter what. The only thing that's changing are the names of the enzymes, okay? So in both groups, Mut-S and Mut-L recognize and cut. Over here, you're going to have RPA and RecJ, RPA in eukaryotes, RecJ in prokaryotes, physically removing. And then you're going to have DNA pol delta or DNA pol 3 come in and resynthesize. And then in both species, a DNA ligase is going to come in. They are technically different enzymes, but they do the same thing. DNA ligase comes in and it's going to fix the backbone. And now you've repaired that mismatch. But what happens if we get a different type of damage? So that's damage that's going to happen when you get a mismatch during replication. Another problem that we may run into is when we have damage that comes from UV light or we get damage from reactive oxygen species. So these two types of sources of damage, so UV light would be an exogenous source, right? It's usually from the sun or exposure to some type of UV light source. Or reactive oxygen species, this is an endogenous exposure. For the most part, I guess theoretically you could be exposed to reactive oxygen species in the wild. Cigarettes, for example, do put a ton of reactive oxygen species into your body. Can damage DNA in a couple different ways. And by the way, if you're curious about the different species of reactive oxygen species that we encounter in our body, right here. Oxygen is fine, but superoxide, peroxide, hydrogen peroxide, hydroxyl radicals, hydroxyl ions, these are all things that can actually damage cells. Particularly with UV light, you're going to end up with something called thymine dimers. It technically can happen with any pyrimidine, but most often is with thymine. And essentially what happens is you can see you have two thymines, two T's right next to each other in the DNA sequence. The UV light comes in, and instead of consisting of two separate nucleotides, these two thymines now fuse together, and they are now covalently bonded outside of the covalent bond that's supposed to form the backbone. So these two bases are now glued together. And because they're glued together, they can no longer base pair with the neighboring strand. Okay? So now you essentially have this like non-paired bases in the middle of your DNA. And you can't really easily break them apart because these two are now essentially glued together, right? You now have a covalent bond between these two. And as we've talked about, bases are connected with hydrogen bonds, easy to separate. These two nucleotides are now stuck together through covalent bonds, and now you need an enzyme to fix this. And you don't really have an enzyme that can allow that to be fixed easily and get rebase pairing. Another thing that can happen are the formations of what we call bulky adducts. So you can see here's the nucleotide here, but the damage is these additions of these giant bulky adducts to the side. And here's the problem with this. These bulky adducts are going to fully prevent hydrogen bonding from occurring between bases. So either way, what you have is two different ways of disrupting base pairing within the nuclear or the DNA duplex. And that's highly problematic. And the only way to repair these is through something called nucleotide excision repair. So what you can see here, and I'm showing this here, we'll talk here about, we'll just say we're talking with thymine dimers. So you can see the effect, right? You know that these T's and A's are supposed to be hydrogen bonded. There should be two hydrogen bonds that exist to connect these two. But because these two thymines have been paired through UV radiation, you've now got that dimer, they can no longer connect, all right? Once the dimer is detected, the DNA is going to get opened up to form a bubble. And essentially what we're going to do is kind of like with base excision repair, we are going to remove the whole chunk. Okay, that whole chunk comes out. We put a whole new chunk in, and then DNA polymerase replaces it, and ligase seals the backbone. Okay, now nucleotide excision repair can come in a couple different varieties. It could be through transcription coupled repair. So, for example, this may not get caught in a normal cell. This might get caught during the process of transcription, right? So they're trying to transcribe. They see this sort of mismatch, this mess with these thymines. Obviously, transcription can't happen because you can't get the base pairing needed for RNA polymerase to do its job. And it goes, hey, there's a problem. The damage is recognized and this process begins. Or it could be recognized following the damage, right? So when a cell is exposed to damage, it kind of sends out these DNA repair proteins to be like, hey, something bad happened, go take a look, right? It's like your security alarm goes off. You don't go, oh, well, the alarm stopped going off. I guess there's nobody in the house. Somebody's got to check the cameras. Somebody's got to go make sure that nothing bad happened. So it can be detected here as well. And what's going to happen is you get a series of enzymes come in. So XPG, RPA, XPE, as well as, in the case of transcription coupled repair, this is going to be detected by the transcription initiation complex. That's why you have transcription factor 2H here. They recognize it. They bind. They unwind the DNA helix. All right. They then cut out the strand. DNA polymerase comes in, fills the gap, and the ligase seals the nick. Okay. It's a very similar process to what we've seen with base excision repair. So very, very similar there. It just involves different proteins, and it's slightly different at the molecular level. And either way, you get a clean template for transcription. So if this was detected during transcription, you're good to go. If this was detected during the replication process, say, for example, DNA polymerase goes, hey, I can't base pair these T's because they're dimerized. Well, Either way, depending on how you detect it, this comes in and we get a clean template for replication or a clean template for transcription, depending on which process we are going through. X-rays, reactive oxygen species, and alkylating agents can cause a series of different problems as well. So X-rays are a form of highly powered radiation that can damage cells. We talked about reactive oxygen species. In addition to causing bulky adducts, they can cause some other things. And then alkylating agents. So these are things like nitrogen mustard and other like toxic chemicals that you can be exposed to. These can damage DNA in various ways. So, for example, you can end up with an abasic site. In other words, the damage quite literally just knocks an entire base out. So you don't even have a G here to base pair with the C. It's just a gap. You have an unpaired cytosine here. You can get deamination. What ends up here is essentially with the uracil, you remove some of these nitrogen-containing groups. And the problem here is without these nitrogen-containing groups, you don't have base pairing capabilities. You're losing where the hydrogen bonding is meant to occur, right? Because you need to have that nitrogen bound to that hydrogen in order to get that polar covalent bond that allows hydrogen bonding to occur. So that can disrupt base pairing. In some cases, guanine can be damaged. So you end up, instead of having guanine, you have 8-oxoguanine. And you can see here, this is the normal base pairing, 1, 2, 3 hydrogen bonds. But with oxoguanine, you get 1, 2 base pairs. This is going to result in a problem. And in a very bad case scenario, you end up with single-stranded breaks. In other words, you've actually broken the entire backbone of that molecule. So any one of these obviously causes major problems with the DNA. You're not going to be able to replicate this appropriately. You're not going to be able to transcript from this appropriately. So how does this get fixed? This is, again, going to get fixed through base excision repair. All right. So what ends up essentially happening in many of these cases is you end up with deamination. Sometimes deamination can lead, for example, a cytosine to convert to a uracil. That's an example of the damage that can happen. Okay. So you end up with base excision repair coming in. In these cases, the way to fix this is you essentially remove the nucleotide that's a problem. Just that one. Notice we're not removing an entire stretch of DNA, right? We're just removing that single base. And in fact, we basically create an abasic site. So if we have an abasic site, this is where we're starting from right here. Okay, we have a nucleotide minus the base. The baseless nucleotide gets removed. Okay, so pop it out. And then all we have to do is use DNA polymerase to come in, make the right base pair, and seal the gap with ligase. That simple. Okay, so that's one mechanism for base excision repair. And you can see what's happening right here. It depends on whether it's going to be a short patch or a long patch. Cells choose different ways of doing this. It depends on the nature of the damage. This is a very close example, very simple example of short patch. You get that problematic base. It gets removed. You come in and you essentially repair the one nucleotide. Okay. In some cases, it might be easier to go through and cut out a longer stretch of DNA, and then you basically come in to create a flap. You basically lift up several bases. You peel it back a little bit. The flap endonuclease comes in. Just so you're clear, an endonuclease is an enzyme that can cut DNA, removes that, and then it comes in and it repairs it with, again, DNA polymerase and DNA ligase. So in the case of short patch, it's going to be pol beta and DNA ligase 3. In the case of long patch, you're going to need pol beta, delta, and epsilon to come in, and then DNA ligase 1 fixes it. Okay. For the most part, this is going to depend on the nature of the damage. It's going to also depend on which type of cell it's being repaired in. All right. Again, X-rays, UV light, and reactive oxygen species. These can cause different kinds of breaks too. One of them is called a double-stranded break. This is highly problematic. You've literally severed an entire stretch of DNA. You've cut it in half. Okay, we'll talk about why that's a huge problem in a minute. Or an interstrand crosslink. And what's happening here is, remember, hydrogen bonds are meant to join bases. With an interstrand crosslink, what ends up happening is you covalently bond the bases now. Now you have a covalent bond holding together the two strands of DNA. That can't be separated through the action of transcription factors in the case of transcription. It cannot be separated by helicase in the case of replication. You now have two strands of DNA that are physically glued together at a certain point. So either we're completely cutting these, completely severing the two strands, or we're gluing them together. And either way, this is going to require a type of repair called recombinational repair. And there are two ways that this can happen. One is through a process called non-homologous end joining, and the other is through a process of homologous recombination, okay? But which one depends? Where are you in the cell cycle and what's available as a template, okay? So in both cases here, we're going to examine a double-stranded break, okay? Non-homologous end joining directly ligates two broken strands together. It also uses compatible overhangs to prevent mutations. All right. So what that means is you can see here with this double strand break, it's not linear. It's not straight through like they're showing here. You can see there's a little bit of this strand overhanging, a little bit of that strand left. What that means is this is probably the overlap for this and can serve as a template to synthesize this missing part. This little bit here can serve as a template for this strand and can be used as a template to synthesize this. This is fine unless you don't have any overhangs available, which you're seeing here. The problem then is in many cases with non-homologous end joining, if you do not have any of these overhangs available, it's going to kind of just mash the strands of DNA back together. And the problem with that is it can be mutagenic because you don't know if there's just like, imagine, like we're showing this right here. We're just assuming that this base and this base are supposed to be the neighboring bases. What if this chunk is obliterated? What if there are like 4 nucleotides in here? Well, when we just glue these ends back together without asking any questions, those bases are just gone. Like they're gone. So whatever information is missing is now just a deletion. They don't exist anymore. And if that happens inside of a protein encoding gene, now we have a real problem with that resulting protein, right? We have a massive deletion at this point. This is going to predominantly happen during G1 of the cell cycle. So if you remember our talk about mitosis and meiosis from my previous videos, one of the things you'll remember is G1 checkpoint is kind of like a big deal. This is where a cell is going to look around and go, okay, is my DNA in good shape? Do I have a lot of mutations anywhere? Excuse me, do I have any mutations anywhere? Any major damage to the chromosomes, anything like that? If it detects a double-stranded break due to some type of radiation or exposure to a chemical, it's going to try to fix it through non-homologous end joining, okay? Because there's nothing else available to it. And that's the thing. Right here, we only have this particular duplex DNA as a template. We don't know. But once we've gone through S phase, if you recall, now we have homologous chromosomes around, right? So we do have homologous chromosomes, right? We have mom's chromosome 1, dad's chromosome 1, mom's chromosome 2, dad's chromosome 2. But these haven't been duplicated yet. So we can't rely on those as sources. But during S phase, during G2, during the stages of mitosis and meiosis, now we have 2 copies of dad's chromosome. We have 2 copies of mom's chromosome 1. Now we have additional information in there. And what that means is this. About a third of the time, that sister chromatid, remember the exact duplicate, that sister chromatid can act as a template because it goes, oh, I've got another copy of this laying around. Let's go find out what that copy says. And essentially what happens is that sister chromatid loops out, acts as a template for that stretch of DNA and says, okay, this is what that other version of the chromosome says. That's what this one needs to say. So you're using an existing chromosome where you know you have that information and saying, oh, okay, I'm just going to duplicate this. But you have to have that available. 2/3 of the time, it's a homologous chromosome. So it could theoretically be using mom's chromosome 1 as a template for dad's chromosome 1, which understanding is not necessarily mutagenic in the sense that you're not going to get an insertion or deletion, but you may get a bit of mom's chromosome 1 on dad's chromosome 1 if that's how it's being used. Okay, so the key here is you're no longer relying on the duplex strand. You're no longer saying I've got 2 strands of DNA. I hope there's a bit of overhang so I know that this is supposed to go back together like this. Instead, we are using information from a whole different chromosome, but we're looking at that section, the homologous section of that other chromosome and going, oh, I see. So you fell asleep in class, you missed a minute of notes, you go to your neighbor and go, hey, what did he say during that minute that I fell asleep? And you copy it down. I mean, you're hoping it's the good information, but at least here it's not going to be mutagenic because at least you had that information. The proteins involved are different depending on which type of recombination repair you're doing. What happens, on the other hand, if we get an interstrand crosslink? Okay. Typically, we're going to rely on a series of nucleases to come in. They're going to have to cut out that section of the chromosome or of that particular DNA. And then typically they use homologous recombination to be able to fix this. Okay. If you don't have homologous combination available, there's something called the Fanconi anemia pathway, the FAT pathway. If that's not available, then you can use non-homologous end joining. The problem here is that non-homologous end joining isn't a very good process for doing this, and this can lead to cell death. One of the ways we actually use this to our advantage is we use chemicals to treat cancer cells that cause intentionally these interstrand crosslinks, because even though there is a way to fix this, it doesn't work out often, and usually it stalls out and the cell basically goes, I can't fix this, and it goes through the apoptosis pathway. So when you get these interstrand crosslinks, the cells usually detect them, and the end result in many cases is the cell's going to end up dying, but that's a good thing, right? You have a non-functional chromosome. That cell can't possibly be good. So knowing this, we've developed anti-cancer drugs to specifically cause this to happen in tumor cells because they can't divide, and as a result, they just end up dying, or at least not dividing. So it's a good way of doing this. The problem is, just remember, when it comes to cancer therapeutics, all of our cells are essentially the same. So we want to make really sure this is only impacting tumor cells, because it can cause cell death in healthy cells too. And that's one of the reasons why chemotherapy is so harmful. There are so many side effects to chemotherapy in the patients that are undergoing chemotherapy for cancer. So regardless of the DNA, right, regardless of the type of mutation, regardless of the repair pathways used, if a mutation goes without being repaired, okay, if it goes without being repaired, that mutation is going to alter the DNA sequence. Okay, so like we talked about, mutations, they can occur from endogenous sources, DNA replication errors, reactive species, or exogenous exposures. So things like exposure to UV light, exposure to radiation, X-rays, certain carcinogenic chemicals can have that. The type of mutations we get in the DNA depend, right? So you've got point mutations. This can happen if mismatch repair doesn't happen appropriately, right? That daughter strand now includes the wrong nucleotide. And then when it goes through the next round of replication, the wrong nucleotide is going to get base paired with it, and now that's permanent. We talked about ways in which insertions and deletions can happen, right? We get that strand slippage in highly repetitive areas. It's not always how it happens, but that's how it can happen. It's a major way in which it happens. And the result is an insertion. In this case, we get an additional nucleotide. Notice it's a C going right next to a C, right? Strand slippage. Maybe there was a bunch of G's on the other side and this got inserted. Or a deletion. We get that strand slippage, and now the daughter strand doesn't incorporate that new nucleotide. And occasionally we get substitutions where a whole chunk of DNA gets replaced with something else. This, for example, could be an error in homologous or in non-homologous end joining, right? Now, what's interesting is, what are the consequences of these mutations? Okay, because let's just assume we get through all these repair processes, and now this nucleotide is now or nucleotides are just incorporated in all subsequent versions of that DNA, right? Once it happens, it's there. And any DNA resulting from the replication of that new daughter strand is going to incorporate that new mutation. It's now permanently affixed in that particular organism and all subsequent organisms. Mutations in single-celled organisms. Remember, there's only one cell. They're going to reproduce asexually for the most part. Basically means if this mutation exists, all subsequent generations resulting from that particular cell's division are going to carry that mutation forward. And evolutionarily speaking, in single-celled organisms, that is where genetic diversity comes from. It comes from that mutation and just getting passed down because there's no gene pool, there's no sexual reproduction. In sexually reproducing species, this mutation has to happen in a gamete, right? So if this happens in one of our skin cells or a liver cell or a lung cell, we might get cancer, right? That can happen, but that can't be passed down to the next generation. If it's going to happen in a sexually reproducing species and matter for the future of the species, it has to happen in a gamete, a sperm or an egg. And that's one of the reasons why when you go get x-rays, for example, they tend to cover up your sexual organs with one of those lead aprons, right? They're trying to protect the testes or the ovaries where the reproductive cells are or are being generated so that you don't damage the gametes. Because the thought is if you damage a woman's egg cell, and that egg is the one that gets fertilized, that mutation is going to make it on to the next generation and could end up in unknown damage, right? Could mean nothing, could end up in stillbirth, or it could end up in some type of inherited illness. All right, so just keep that in mind. If it happens in a single-celled organism, it's automatically impacting the next generation because there are no alternatives. If it happens in a multicellular organism that reproduces sexually, it's got to happen in a gamete if it's going to impact future generations. So I know we've talked about this, but I want to remind you all of why this matters, right? Because if we mutate the DNA and if it happens inside of a gene, then when that gene gets transcribed, the mRNA is going to be wrong, or I shouldn't say wrong, it's right. It's just different than what we would call wild type, different than the original version of that particular gene. And then when it goes to the ribosome isn't doing any proofreading. It's just looking for the right tRNA to match the right codon, right? And if that codon's off, you might end up with the incorporation of the wrong amino acid. You might end up with incorporation of no amino acid in a stop codon. You might end up with a frame shift in a series of wrong amino acids. I talk about that in my mutations video, so I'm not going to go down this rabbit hole. But what happens if a mutation doesn't occur within a gene? Does that necessarily have an effect? And how do cells handle that? So just a reminder that not all of your genome is comprised entirely of genes. So if you're a prokaryote, if you're a bacterial cell, your chromosome is highly gene dense. You don't have a lot of room in your chromosome that isn't encoding some type of gene. So that means if you get a mutation, it's probably going to be in a gene or in some type. If it's outside of a gene, it's probably in a regulatory element, right? So like a promoter region, for example. But if you look at a human genome, which we're showing here, look at this pie chart. Only 2% of our genome actually encodes protein encoding genes. So that means there's like a 98% chance of a mutation not even impacting a gene. But is it possible for a mutation to impact the expression of that gene or a series of genes if it doesn't actually land in the gene? And the answer is yes. Okay? Mutations that occur in introns are likely not to impact gene expression, but they can affect the proteins. So if you think about it this way, you've got kind of you got kind of a couple of categories. You got a few categories here. First off, you get a mutation that actually impacts the gene. I've talked about that in my mutations video. That's where we get into things like frame shift and nonsense versus missense and silent mutations. Okay. What happens if it lands in a eukaryotic gene, but it lands inside of an intron? Well, it's probably not going to screw with the expression of that gene, but depending on where it is in the intron, it can affect that protein. So it's not technically inside the protein encoding part of the gene, but it can screw up the protein. We'll talk about that in a minute. Or you could get a mutation that occurs inside of one of the promoter regions. And again, promoter regions are big. They can tolerate some mutations, but if it happens in a very critical region of that promoter, it can make it so that gene doesn't get expressed, or it gets expressed too much, or it doesn't get expressed at the right level. And then you've kind of got like the remainder of this pie here. You've got like all these lines and signs and retrotransposons and segmental duplications and all this. And the chances are if it lands in one of these areas, it doesn't matter. It's just not inside a regulatory element. It's not inside of a gene. It's not inside of an intron part of a gene. It's just not a problem. So it's probably not going to be an issue. So like I said, genomes are not entirely comprised of genes, and mutations that occur outside of a gene have a reduced likelihood of affecting gene expression. But what happens if a mutation lands inside of an intron? So recall, eukaryotic genes have two different parts. You've got exons, or expressed sequences. These are the parts of the gene that will encode the protein. And then you have introns, or intervening segments. And these are the part of the gene that need to be removed through splicing of the mRNA prior to translation happening. So recall, eukaryotes have all that processing that has to happen after transcription. You got to get your poly A tail, you've got to get your 5' cap, but more importantly, you've got to go through the process of splicing. And if you don't do the splicing, you're going to end up with gaps. You're going to end up with all of these segments included that don't incorporate the protein. Now, for the most part, if a mutation occurs inside of an intron, there's a very low likelihood that it will affect the resulting protein, unless it happens at an intron-exon boundary. So if you look at an mRNA, in eukaryotic mRNAs, the border of an exon and an intron always has a GU at the 5' end and an AG at the 3' end. It's just a rule. We don't know why. That's just the way evolution decided that. If you damage the sequence on either shoulder of this intron, it may make it so that the spliceosome is not able to remove that intron. It'll remove all the other introns, but that one stays. And as far as the spliceosome is concerned, I didn't have the sequence to cut this out. I'm treating it like an exon, and we leave it in play. And what you've now created is a very large insertion into the mRNA, and now you're going to get that being translated. Now, what's the impact on the protein? Either way, it's not good because you got a bunch of amino acids that shouldn't be there. So that's absolutely going to affect the shape of the protein. But also, if this is not a number that's divisible by 3, if that intron is made up of nucleotides, a number of nucleotides that isn't divisible by 3, you're getting a frame shift as well, right? Because it's going to go 3, 3, 3, 3, and it's got to be back on 1 when it starts at exon 2 here. It's got to be back on 1, 2, 3 to start that next exon. If it's not, now everything past that insertion is out of frame, and now everything's going to be wrong. So you can see why, even though this mutation happened in an intron, it happened at a very critical region of that intron that prevents that intron from being removed, and as a result, gets treated like an exon and just gets encoded as in the resultant protein. Hugely problematic. What happens if it's completely outside of the gene, right? If it's completely outside of that gene, what happens, right? So the problem here then is that if it happens in one of the promoter regions or one of the enhancer regions upstream of this, well, transcription factors require specific DNA sequences to bind. If those sequences get mutated, okay? So again, it's not the entire promoter, it's not the entire enhancer, but there are certain sequences that promote that transcription factors have to recognize or they won't bind to the DNA. So if those sequences get mutated, the transcription factor won't recognize the enhancer. It won't recognize the promoter. And as a result, you might get no expression of a gene. You might get too much expression of a gene. You might get limited expression of a gene. It depends. Which sequence is being disrupted, which transcription factor or factors are being impaired, and that can have a huge effect on the expression. Now, the gene being expressed is fine. The protein is not mutated, but you might get too much of it or too little of it or none of it at all. And because proteins are largely the doers within a cell, this could be a situation where all of a sudden we're just always, we call it constitutively, expressing a gene or a protein that's responsible for cell division. So if we're just always expressing a signal that says divide, 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 you may have basically made this cell prone to cancer because it's always got the gas pedal down for division. Or on the other hand, it might break something that's, for example, one of our tumor suppressor genes. One of the genes who normally functions to turn off the gas. It's the brakes in the cell cycle. This one's not getting expressed at all of a sudden. Again, the protein's fine, but it's not being expressed because you've disrupted the regulatory elements that control its expression. So all of this can be problematic. Okay? Going back to what happens if it happens with a gene, again, I've covered this in other videos, but again, we can get point mutations, right? Point mutations can lead to silent mutations in the protein, missense mutations where we get an amino acid substituted for another one, or a nonsense mutation where we get a stop codon in place of amino acids. Okay? Again, we've talked about these. I'm not going to go through these. Or insertion and deletions, they're going to give you that frame shift that we've talked about. Okay? I will note we have no ability to determine how that's going to affect protein activity just by looking at the sequence. We can take some guesses, but we can't definitively say what's going to happen. One of the ways that we can guess whether a mutation, so let's just say we get a missense mutation, right? A silent mutation is going to have no effect. We're not going to worry about that. A nonsense mutation is going to terminate the protein early. We can probably assume that bad things are going to happen. But what if we get a missense mutation? Where we get an insertion and deletion at a certain point? Is there a way for us, okay? Is there a way for us to guess or to have an idea of whether or not this mutation might impact protein function? And the answer is actually kind of yeah. Because if we've studied a protein enough, if we've studied a protein enough to know like sort of where its domains lie, what it does, how it looks inside the cell, where it functions and how it functions, we might be able to guess whether a mutation is going to happen. And the reason why is all proteins have critical regions. Some of these critical regions are regulatory domains. These are places where post-translational modifications happen. Maybe it's phosphorylation, which activates the protein, and then the subsequent dephosphorylation turns it off. Okay, maybe it's a spot where methylation occurs that activates it or deactivates it, right? We could get dimerization domains. So, for example, some proteins need to come together in order to function. So, for example, hemoglobin requires multiple subunits coming together. Well, if you prevent that from happening by mutating one of these dimerization domains, that protein can't form its tertiary structure and it won't work. If it's a transcription factor that needs to bind to DNA, or even if it's like a DNA or an RNA polymerase that needs to bind to DNA, if you mutate the spot required for DNA binding, that protein can't function any longer. These transmembrane domains, these little stretches of usually non-polar amino acids, if you introduce, say, a polar amino acid in here, it could make it so that this part won't insert into the membrane appropriately. This will cause the protein to misfunction and not be able to sit right in the membrane. If this is a channel protein or this is a receptor, it might behave not at all or erratically because of this. If it's an enzyme, you've got that defined active site. That's where the enzymatic activity happens. If you mutate the active site, it may not be able to fit its substrate or perform whatever it does, whatever duty it does to cause the catalyzation of that particular reaction, right? Or we've got binding pockets in receptors. So receptors have to fit their ligand appropriately. Well, you can imagine if all of a sudden there's like a bumpy part sticking out in the middle, its ligand can't fit. And as a result, that receptor just goes, oh, I don't recognize my ligand. It doesn't bind appropriately. And you don't get the transduction of that signal and then the downstream effect that you're looking for within the cell. So let's look at an example or two. The first one, CFTR. So CFTR is the protein that is mutated in cases of cystic fibrosis. So there are a bunch of different mutations that happen here, okay? And what they've done through research on cystic fibrosis is created different classes based on how the mutation impacts the protein. So there are about 2,000 different mutant alleles that have been discovered, which means different mutations in the DNA. But they all kind of result in very similar, they all kind of condense on a very similar, like a smaller group of mutations in the protein. So, for example, with class 1 CFTR mutations, you end up this X right here, this G to X, W to X. So, by the way, just in case you want to know how to read these, The G is the amino acid. In this case, it would be a glycine at the 542nd position. So it's the 542nd amino acid in this protein. It should be a glycine, and you get a nonsense mutation. It stops right there, truncates the protein. Here you've got a tryptophan with a stop with a nonsense mutation at amino acid 1282 or at amino acid 553, so right next to this glycine. There's supposed to be an arginine if you get a stop mutation here. All of these result in no functional protein being made. Essentially, people with these types of mutations do not produce CFTR at all, which results in no chloride transport. Class 2s, similar mutations. You can see you get a deletion here, a deletion here. This is a missense mutation. This is an asparagine being converted into a lysine here, NK. But over here, these are deletions. This amino acid is just missing, okay? They get a CFTR protein, it's misfolded, and it doesn't ever make it to the plasma membrane, so you get no transport. Here you get one that does fold, does reach, but doesn't function appropriately. In class 4 mutations, the opening is faulty, so it doesn't open right. And over here, it does work, but it works poorly. So what you're seeing is these are the examples, okay? These are the examples. Here, what you can see with this one, this right here is a 10 kilobase insertion. There's 1,000 nucleotides that aren't supposed to be there, and it's there. Here you get 5 guanines added, just right in here, and they become A. 5 of these Gs then become an A, and that causes mutation. And here you get an alanine becoming a glutamic acid right here. So that's just a missense mutation. The end result is either a non-produced CFTR, a produced one that misfolds, or don't function appropriately. So the bottom line is all of these different mutations in one way or another impact chloride transport through CFTR. The end result is to mutate the protein in such a way that it can't function. I did misspeak, so I'm going to go back and I'm going to correct this right now. Class 5 mutations are not within the protein for the most part. These right here are referring to regulatory regions. These mutations impact the promoter, which means the gene doesn't get produced in sufficient quantities to make enough protein, resulting in a decreased amount of chloride transport. So I'm wrong. These right here are not within the gene. These mutations up here are within the promoter region. And as a result, you get a mutation that causes a lack of sufficient production of the protein. So this kind of goes back to this other one, right? This is back here. These ones are outside of a gene. The remainder that I talked about are within the gene and cause the protein to be defective in some other way. Hemoglobin is another example. So in hemoglobin, all right, you can get a single nucleotide point mutation in the beta globin subunit. Okay, so you've got alpha globin and you've got beta globin. And in order to make an appropriate hemoglobin molecule, you need 2 alphas and 2 betas. All right, this single thymine to adenine point mutation causes a missense mutation where a glutamic acid becomes a valine. Okay, so it should be pro-glu-glu. Here you get proline valine glutamine, glutamic acid, and you end up with this single problem. The problem then is in the deoxygenated state. So when oxygen isn't present on the hemoglobin, it can cause the hemoglobin to aggregate. They stick to each other too much. And as a result, the whole red blood cell actually changes to this sickle shape. This can lead to something called sickle cell anemia. And the reason why is this. These sickle-shaped red blood cells can no longer transport gases. No carbon dioxide, no oxygen. They're kinked in a way. And the other problem is these kinked red blood cells can also block blood vessels. So what ends up happening? Well, because this happens so frequently, it reduces the lifetime of the average red blood cell by about tenfold. Most red blood cells will circulate in your bloodstream for about 40 days before they get removed. Unfortunately, people with these sickle cell shaped, the average lifespan of a red blood cell is now 4 days, which puts a huge burden on the body. So essentially, you have to produce 10 times more red blood cells than average people. Well, red blood cells consist of hemoglobin. Hemoglobin requires iron, and iron deficiency manifests as a disease called anemia. So two things happen here. First off, you can end up with these sickle-shaped red blood cells blocking blood vessels. This can cause a sickle cell crisis, which can theoretically be fatal. The other problem is you're really stressing the system. So people with sickle cell need to get way more iron or risk becoming anemic, which is also potentially fatal because it can make it so they can't produce red blood cells in high enough quantity. Not to mention iron is important for several other processes in your body. So the damage here is twofold. And what's interesting is it's caused by a simple point mutation. One thymine becomes an adenine. The resulting protein gets a valine instead of a glutamic acid, and boom, all of a sudden, we're done. And you end up with this particular phenotype known as sickle cell anemia. What are some effects on an organism? Well, the other thing we have to think about is from an evolutionary standpoint, right? So you can imagine that evolution might want to be like, well, why aren't we making perfect polymerases? Why didn't evolution give us perfect polymerases? Why isn't the proofreading capability of DNA polymerase better? Why don't we have better ways of repairing mutations? Well, the answer is kind of simple. That actually wouldn't benefit evolution. Like there's probably some pressure on there being mutations because mutations are the source of natural variation within a species. All variation within species comes from mutations. When it comes to single-celled organisms, it's really the only source of variation. With sexually reproducing species, you do get a gene pool, which means these variations can kind of hide and accumulate in different ways. That's a conversation for a different day. But when we get different variations of a gene as the result of a mutation, we refer to these as alleles. So when we talk about Mendelian inheritance, we talk about things like dominant alleles and recessive alleles and incomplete dominance and complete dominance and codominance. Well, all of those alleles, these different versions of the gene, are the result of mutations. So mutations are necessities for evolution to occur. Now, the good news is, in terms of evolution, the overwhelming majority of these mutations are going to be evolutionarily neutral. I just told you in humans, for example, there's a 98% chance that a mutation is going to occur outside of a gene because only 2% of your genes encode for genes or proteins, I'm sorry. And even if it happens within a gene, it might happen in an intron and be unimportant. It might happen in a non-critical region of the protein and not be important. So neutral mutation theory teaches us that over the vast majority of mutations will not matter at all. They will have no impact on the survival or reproduction of the individual who possesses them and thus are rendered evolutionarily neutral. And if a mutation is non-neutral, if it actually does have an impact, it's probably going to be harmful. And this is where natural selection comes in. Natural selection, which is largely dictated in terms of environmental conditions, are going to decide which mutations are beneficial and which ones are harmful. And harmful mutations will be actively selected against, okay? It doesn't mean they will disappear from a gene pool. It just means they will be selected against, and that very, 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 very tiny fraction of mutations that actually confer some type of reproductive fitness benefit to the individual will begin to accumulate. And again, all of this is, we refer to this as being relative. So reproductive fitness is relative. It's dictated by the conditions within the environment. And it can change, right? What's good today might be bad tomorrow. What's bad today might be good tomorrow. That's why gene pools are a good thing. It's probably why sex still exists, even though it's highly problematic. But these beneficial mutations are the ones that will steadily be enriched by natural selection. And over time, this can lead to adaptation, okay? And these alleles are the basis for Mendelian inheritance. So when we talk about Mendelian inheritance, we have to recognize that these different alleles, big R and little r, they originated through a random mutation event that failed to be repaired or failed to be recognized, okay? So we do think of mutations as a bad thing. Overwhelmingly, they don't matter. But when they do matter, they're probably bad. So it's not necessarily hard for us to recognize that mutations are bad. Living things are complex, and if you damage a complex structure, it probably isn't going to make it run better. But in some cases, it does. Sometimes Slamming your computer monitor makes it work better. Sometimes hitting your TV makes the picture come in clearer. And that's what happens in nature. Sometimes a mutation can actually benefit an individual. And that's what these alleles are, and that's what mutations function on. So keep that in mind as we move on to future conversations about inheritance and alleles and how that affects the survival of an organism. So keep that in mind for our future conversations. So that's the end of our conversation on DNA repair mechanisms and the consequences of mutations. So keep that in mind moving forward. Thanks for watching, and I'll see you all soon.